First thing I should say is that much of the work that I'm going to be talking about has been done by Ruth that's in the audience here. We've had a lot of help from several of your colleagues within this section, whose names I won't read out, but you can see there. The second thing I should say is that, being an old idiot, I prepared this talk for a rather longer time slot. Hopefully, I can say what I want to say, but if I skip a few slides, please don't have your brainwaves going funny. Right, so I need to start off by reminding you something about the vestibular system and what we're talking about. So the crista, utricular macula, and saccular macula are covered in hair cells, and these tissues are made up of hair cells with intermediary support cells between each one, two types of hair cells, supporting cells sitting on a basement membrane, and between each hair cell, supporting cells between each hair cell. So there's two sorts of hair cells, and there's neurons that innervate the hair cells. That's vulnerable to a number of the hair cells are vulnerable to things like gentamicin here, where you get a loss of crest across the top of the cristae in the striatal region of the utricle, and somewhat less than the saccular macula. There's a sort of differential sensitivity between the crista, utricle, and macula. With gentamicin, or with aging, this is some stuff of Tony Wright's from back in the 80s. So this is well-fixed material. This material, and he tells me a story of how he did this, but these were all fixed within about an hour and a half of death. So this is a 17-year-old male. The crista's covered in hair cells, and in a 70-year-old male, there's very few hair cells left. In the work we've been doing with all our colleagues across the country, this is utricular macula. This is a 65-year-old male and an 80-year-old female. As you can see, there's practically no hair cells at all in this utricle. There's one hair cell there, but there's very few there. So aging can result in a huge loss of hair cells from the utricles. Aged mice, interestingly, also lose hair cells, but the interesting thing here is actually humans are worse than mice in this. These are sort of like 2.5. These are mice about to die naturally. So these are 2.5, 3 years old, between 2.5 and 3 years old. And there are this loss of hair cells, but it's not as extensive as it is in people. This section I'm going to gloss over there. I may skip some slides here, because the first thing I was going to talk about is one way to prevent this loss is to use various sorts of protectants. We know that the hair cells died by apoptosis. This is some work we did a long time ago, and these are hallmarks of apoptotic cells. And we did some experiments that said if you block the enzymes involved in generating apoptosis, which we won't worry about that one, means things like this, which are pan-caspase inhibitors. This is a control number of hair cells. This is gentamicin alone. These are all from utricle, two different species. You can protect hair cells from loss. So hair cells can be prevented from dying. This is a concept that has gone on to produce a number of different things, which I won't go through. This was a nice experiment Ruth did doing. Oh, well, I will go through this one because I like this. This is one of the nicest experiments we ever did. This is actually organ of Corti, but it's a principle. You take the organ of Corti out, you drop it in a dish. Green are cells, this is live, so green are cells which are alive, red are nuclei of dead cells. You can see if you take the outer hair cells from the apical end, they're all alive, they've died here. If you take the same tissue, you take the basal end, and you know with gentamicin there's a base to apex gradient and sensitivity. Apical hair cells are all right. After 6 hours, all the outer hair cells are dead. Similarly, inner hair cells, which also survive longer, are alive when outer hair cells are dead. So most of the outer hair cells are dead, most of the inner hair cells are alive. So this pattern, just by dropping something in a dish, you can replicate that effect. And all you do is you add some free radical scavengers. All of these are different free radical scavengers, salicylate, mannitol, glutathione, and so on. Paraphenylene diamine, which is something we picked up off the shelf. You can allow cells which would normally die at this rate are protected. That's led on, a lot of other work. I mean, it's not the only piece of work done this, but it's just a very simple experiment which demonstrates a fundamental point. That you can use various agents are now going into clinical trial, which Ralph referred to earlier, to try to stop hair cells dying. Now, that's a concept, that's something that potentially would work, but if it's trying to stop aging, you're going to take all these things the whole of your life. How long do you have to take them for, how effective they are? And there's a lot of other things that suggest that enhancing free free radicals may not necessarily always be a good thing. And it may not be the only thing you need to do. These are pictures from these are well-fixed samples of older people. These are the stereocilia, and what you can see here are things that my colleague Nico Dowry calls stereociliopathies. All of these are abnormal hair bundles. These are clearly persistent for a long time before the hair cell dies. So the hair cell is going to be non-functional for some considerable period of time before it actually dies. So protecting from damage is not going to give you recovery. And one of the reasons to say that that is not a fixation artifact, and these were fixed again within 2 or 3 hours of death, you get the same kind of anomalies in old mouse. So these are old mouse hair cells showing exactly the same pathology. So that is a pathology of aging. Okay. So if you can't protect them, what do? Is you try to regenerate them. Now, if you're a bird, this is the basilar papilla of a bird. And these are the supporting cells, these little ridges here. Obviously, these are the hair cells. If you ablate the hair cells with gentamicin, all the hair cells come out, the supporting cells close all the gaps up, and within 5 days, you've got spontaneously new hair cells are coming. These are the same magnification. These are baby hair cells coming out within a very short space of time spontaneously. Another animal we've used, which is quite a nice little thing, is the newt. Newt's a wonderful animal for doing regenerative studies. It can regenerate all sorts of parts. I won't go into it. It's sometimes called the champion of regeneration. This is what its saculum macula looks like, which is a hearing organ in newts. It's undamaged. You ablate its hair cells. There's nothing there left. And within about 3 weeks, you have baby hair cells coming back. So these are 2 models we can use to look at regenerative properties. We also discovered some years ago that even in mammalian utricular maculae, you can get a level of regeneration. Normal, ablate the hair cells, and over time, you can get new baby hair cells coming back. You can see that the thing, it's quite a long time. We're taking 12 weeks here. This is not, and the bundles look as little babies. These are little baby bundles. Here's a mature bundle, and they have different morphologies. And the relevance of seeing that picture will come back in a little while. And it's not repair, because if you take sections, this is normal, you ablate the hair cells, and you can see hair cell bodies coming back. So it's not just bundle recovery. The loss is only about, over this period of time, about half of what was lost. You don't get complete recovery. So if you have a pathological situation, essentially what's happening is hair cells are being lost and recovered, and it's going on a downward slope. And those hair cells become innervated. This is all done this is all in vivo. So this is all in vivo in guinea pigs. They become innervated. So regenerated hair cells will become reinnervated. Okay. Now, in birds, there are 2 ways in which have been identified for new hair cells to appear. You take the hair cell, you ablate it, you're left with a supporting cell. The supporting cell either goes into cell division, makes 2 cells and makes a hair cell and a supporting cell, or 2 hair cells, but basically it recovers the number that were lost. Now that's actually quite interesting, because not only do you have to start off and trigger a response which doesn't normally occur, you have to stop it at some time when the right number of hair cells are there. Now nobody knows how that happens. But the other way is the supporting cell turns directly into a hair cell by something called, some people call transdifferentiation. I prefer to call phenotypic conversion. The cell turns from one phenotype to another. Now, in the newt, we did some studies, or Ruth did some studies. This is to get you, so these are, the green are all new hair cells. The red are nuclei which have undergone cell division. And this is different levels, confocal images through the tissue. And the point here, if you look through that, is you will not see a green cell with a red nucleus, and there's no red nucleus in a green cell. In other words, none of these hair cells have been generated through cell division. Supporting cells divide, but the hair cells, at least the initial ones that occur, do not come by a division process. And if you start looking at sections, you can see these hair cells there have a little bit going down to the here, which is normally hair cells don't meet the basement membrane, but here you see cells with extensions down to the basement membrane, which is just what you'd expect if a supporting cell turned into a hair cell without an intervening mitotic event. That kind of process has now been shown to occur in chicks initially before the division occurs. And in mammals, if you have a hair cell, a supporting cell, sitting on a basement membrane but has an organised bundle. A more organised bundle is got a foot plate, and an organised bundle, and there's a foot going down to the basement membrane. So this is morphological evidence that in mammalian systems, you get conversion from a hair cell, a supporting cell, into a hair cell. Now, interesting, looking through our human material from elderly people, this is almost no hair cells there, but look at, where is it? That's one here. You've got a cell there that has some of the characteristics, and we look at these in different elderly people, which have characteristics of immature hair cells. There's a hair bundle from a basal-prepared chick. It doesn't look very different from. And these are from our guinea pig utricles, which don't look much different from either those or those. So this is naturally happening in old people. You can find immature hair cell bundles, suggesting that regeneration is retained within the human utricle throughout life at some level. Now, if you want to manipulate that process, which is what we want to do, you need to understand something about the way hair cells and support cells are produced. And this is just simply to show that you get a cell division to produce a sensory precursor and a neural precursor. That stops dividing, divides nerves. The sensory precursor divides once more to become the sensory progenitor rather, becomes precursor, which then differentiates into one or other of these cell types. So these two cell types arise from the same mother cell without an intervening a mitotic event. They're two alternative fates. Cells that express ATOH1, this gene here, become hair cells, and they inhibit their neighbours from following the same cell fate by activating a system called the Notch delta signaling pathway, which I won't go into. But the point is, cells want to become hair cells. The first cells that do to express ATOH1, and they inhibit their neighbours, and those neighbours become supporting cells. So the two cell types are from the same precursor. They're alternative fates. So if you want to induce phenotypic conversion, you might think of two ways of doing it. One is gene transfer using ATOH1, and the other is to manipulate this Notch delta pathway. So you would basically stop the cell being a supporting cell, and it would go on to become a hair cell. To give that diagrammatically, normally, ATOH1 leads to the formation of this thing here called delta 1, which interacts with Notch, which causes a cleavage at that point, and this Notch intracellular domain interacts with the nucleus, producing HES, which blocks these two proteins, so this becomes a supporting cell because it's inhibited, okay? So the idea would be, A, you transfer ATOH1 into one of these cells, so you overcome this block, or you block this system here. And there are various ways of doing that. There's things called TACE inhibitors, which essentially block at this point. There's DEPT, which blocks this cleavage here, so this bit doesn't happen. So what then happens is you don't get HES1, you don't get a block of ATOH1, so what you would get is hair cells instead of supporting cells, okay? So let's see what happens when we do that. So what we've been doing is taking utricles. Our clinical colleagues have been driving us from surgery for acoustic neuroma, or vestibular swarmanova, whichever name is popular with you. This is an SEM of the whole thing. What we've done in this case is we've exposed it to FM143, which is a dye which will only go into living hair cells. So the hair cells take up FM1. So we know that cultures have living hair cells when we start out. That's a sort of quality control. We can maintain those in culture. Myosin 7 is marking the hair cells. So you can see after 9 days, this in case is in 9 days in culture, you've got lots of hair cells. Supporting cells look good. We've used a number of markers for hair cells. This is one that knows the nuclei. I'm not quite sure how bright that is. I can't see it very well from here. GLAST is something that's expressed by supporting cells. This here, we'll forget about this one, is looking at the way the cells are coupled together. It's a slice. It's a dye is put into one cell. Two dyes are put into one cell, one which can go through gap junctions and one which can't. So that one is confined by the cell, which tells you that that cell's live, because if it wasn't live, the dye would just leak out. And all of these cells are coupled together, and it gives you an idea, just for fun, of you can see what these cells are. This is an area, there are very, very few hair cells in. So this is a culture from somewhere where there are very few supporting cells, hair cells, but the supporting cells retain viability and their ability, as we shall see here, to respond to the loss of hair cells by closing lesions. So here, after 2 days treatment with gentamicin, we've labelled with phalloidin, we have these scars here are where hair cells used to be, and the supporting cells have closed the lesion up. So in all of these systems, the supporting cells are viable. We can get rid of the hair cells with aminoglycoside. We can take these out to 28 days, and essentially nothing happens. These are controls just taken 28 days after gentamicin. And do some cell counts. These are directly fixed. These are various timescales, how many hair cells there are. Treat with gentamicin, and the number drops off over 3 weeks. Okay, so we've got no hair cells. We then transfect the utricle with genes that code ACOH1 and GFP allowing us to see where the cells are. And what you can see is that there are this is the same set of images. This is the phalloidin showing the surface. This is showing cells that are at the surface, i.e. they're supporting cells expressing ACOH1, which shows you how well you can transfect human tissue. So this has a wider significance. If you want to transfect human tissue using these adenoviruses, you're going to be able to do it quite well, actually, and quite efficiently. Any gene you could have transfected. We've transfected ACOH1. That shows the cells that are at the surface. So then we take, in this case, we've used 2 triggers, ATOH1, which you can see from the green, and there are some green cells here, and TAPI1, which is a notch delta blocker. Myosin, that I couldn't find this colour in my palette on the PowerPoints, but myosin 7A is a hair cell marker. So these have been treated with gentamicin. They've then been given treated with this, and what you can see is large numbers of hair cells. So we expected to have none hair cells. So we treated with gentamicin. Using these 2 in combination, you can get very large numbers of hair cells within a human utricle. We can look at those in different ways. If you look at SEM, you can see, this would be normal, that you see all these little things that look remarkably like sea urchins covering things. So you've got all these cells with large numbers of microvilli. Here's a gallery of them, of their features. Now, why that's interesting, again, is this. So there are those 3 hair cell types, and these are hair bundles from regenerating chicken here. That and that will look remarkably similar. That and that look remarkably similar. So we seem to have some evidence here that we can regenerate hair cells, that they bear bundles of a certain kind, and when we take sections through them, and one of the ways we've developed for this is to take SEM samples and then re-embed them so we can cut sections of the same cell. And what we have, if you normally, I'll put this one on first. Normally, if you get rid of the hair cells, this is, and hair cells reduced with no treatment, most of the nuclei are relatively irregular in shape and tend towards the base. Here we have round nuclei, like hair cells, in the upper portion of the epithelium. This one has a round nucleus in the upper portion, and it has all those microvilli on it. So it looks like a hair cell. Okay. So, what we see here is the potential ability to regenerate hair cells in the human vestibular system. And we need to do a lot more on that, looking at the physiology of these cells and trying to enhance the efficiency. But that's the next step going on. But finally, before I finish, there's something else that this has shown. So what we've done here is we've taken the, Ruth's done here, is taken the epithelium off the underlying mesenchyme and put it in a dish. And this is what it looks like after 5 days. Now you manipulate the medium that that's developing in, and they start looking like this. Now this is starting to look like neurons. If you then use markers for neurons, beta-3 tubulin here, you see that cells at a kind of transitional state where they're partly epithelial like this start expressing beta-tubulin, so that when you get these neuronal-like cells, they are labeling with beta-tubulin, suggesting that supporting cells in human tissue, we've done this both with Ruth's done this both with newts and mice, and you get the same sort of result. the supporting cells seem to have this capacity, a plasticity, which allows them to be turned into a different cell type, which is of value. That means there is the potential here that you might be able to induce neuronal, human inner ear neuronal cells from human inner ear tissue, which may have or may not have clinical value. Hopefully, we can then say thank you to the Voice of Research Council, the BBSRC, and Action on Hearing Loss for supporting us. Thank you.